Now coming to sickle cell anemia. Sickle cell anemia is classified under hemoglobinopathies. Under hemoglobinopathies, they are abnormal RBC structures. Okay, so if you see, these are your normal RBC cells, these are your biconvex discoid shaped RBCs. Where you see sickle cell anemia, they are sickle shaped. Okay, so if you see a sickle that you use to cut grass, if you see a sickle, this is the shape. Okay, so your RBCs also take up the shape of a sickle. Okay, so a sickle cell disease is an autosomal recessive disorder. I am sure genetics topic has been taken, so you understand what is autosomal recessive disorder. This is mostly seen in black people of Afro-American origin. Okay, now sickle cell disease is a very old disease. If you go back to evolution, in the Africa, you used to have lot of malaria. So malaria is caused by Plasmodium falciparum. So what happened is, lot of people were suffering from malaria. So by evolution, what happened, these RBCs, the normal RBCs, became sickle cell RBCs. Okay, these sickle cell RBCs were protection against malaria. So by evolution, this change has occurred. So this is mostly seen in Africa. So black people of Afro-American origin. Okay, how this sickle cell occurs is, sickle cell disease occurs due to a partially accepted missense point mutations. Missense mutation refers to a change in one amino acid in a protein arising from a point mutation in a single nucleotide. What happens is, if you see a gene, this is a gene coding. In a gene coding, what happens is, in a one particular point there is mutation. So in a gene coding, in one particular point, if you have a change, okay, this is known as point mutation. And what is missense mutation? When a particular amino acid gets changed, it gets replaced, that is known as missense mutation. Missense means change in amino acids, point mutation means change in a particular location. Okay, so here what happens is, this point mutation results in substitution of glutamic acid by valine in the 6th position of the beta globin chain. So this is a beta globin chain. In the 6th position you have glutamic acid. This glutamic acid gets replaced by valine in the 6th position of the beta chain. Okay, so this will form a mutant hemoglobin S. So mutant hemoglobin S is formed. Okay, so this is a very important thing that you need to remember. Glutamic acid is replaced by valine. Okay, now before we proceed to the forms of sickle cell anemia, I need to discuss the types of hemoglobin. So the types of hemoglobin. Okay, so in normal adult RBC, you have, which is known as HbA, you have alpha 2 and beta 2. So you have alpha 2 and beta 2. Okay, if you remember the diagram, you will have alpha 2 and beta 2. Okay, so and small amount of HbA2, okay, which is alpha 2 and delta 2. Some amount of fetal hemoglobin, HbF, which is alpha 2 and gamma 2. So HbA, you can see over here, HbA is alpha 2 beta 2. Okay, alpha, you have HbA2, which is alpha 2 and delta 2, and HbF is alpha 2 and gamma 2. Okay, so normally if you see in an adult, this is what you see. Okay, this particular structure is what you see. Okay, so when a fetus, especially a neonate, after 6 to 7 months will have an adult type of hemoglobin. Okay, so babies will also have this particular structure. Okay, now You need to know in adults you mostly see HbA, more HbA, 97% is HbA. Some amount is HbA2 and very less amount is HbF. Okay, so these are your types of hemoglobin. These are your hemoglobin that are normally seen in adult. This is only adult that we have discussed. Okay, another point is, when we discuss thalassemia, you need to know alpha globin is seen in chromosome 16. Okay, and beta globin is seen in chromosome 11. Okay, so just remember this. When you see this slide, the things to see is what are the different types. In this, HbA is more. Okay, so in this particular is more. Okay, this particular is seen normally in adults and in babies after 6 to 7 months. Okay, after they are born. So you have alpha globin in 16 chromosome, beta globin in 11 chromosome. Now, forms of sickle disease. What are the different forms of sickle disease? Sickle diseases are of 3 forms. You have heterozygous state, you have homozygous state, you have double heterozygous state. Okay, heterozygous state is known as sickle cell trait. Homozygous is known as sickle cell anemia. Double heterozygous is your sickle beta thalassemia, sickle C disease and sickle 3 disease. Okay, now we will read them in detail. Okay, now heterozygous state. Heterozygous state, that is a sickle cell trait. In this, what happens is, if one parent has sickle cell trait, okay, so this is a normal. If one parent, so this is father, this is mother. Okay, so the father is normal but the mother has a trait. And this, what will happen, not all the babies will get the trait. Okay, so one after one, so 50%. So 50% will get the trait, 50% will get the trait and 50% will be normal. Okay, so there is 1 in 2 chance that any given child will have one copy of HbAs gene and therefore have sickle cell trait. Okay, it is equally likely. Okay, this point is not, it's not important. Okay, now again, if we see, how again can we see sickle cell trait occurring? This is a different phenomena in which again the father is normal but the mother has sickle cell anemia. In this, what will happen? Usually AA will be there. Okay, this means the 2 gene, here the 2 genes will have SS. In this, what will happen if one patient has sickle cell anemia, HbSS, and the other is completely unaffected? So in heterozygous state, what happens? One parent is always normal, other parent is default, is defective. So here again the father was normal, again here father is normal, the mother has anemia. In this, what will happen? None will have sickle cell anemia but all will become trait. Why this happens? Because AS. So the parent who has sickle cell anemia, HbSS, can only pass the sickle cell hemoglobin gene to each of their children. So only the S gene comes into play. So everyone will have trait. So 100% trait. What does trait means? These are carriers. So these are your carrier states. Okay, traits are your carrier states. Okay. Now coming to homozygous. Homozygous is more what? Both are affected. Okay, and this both parents are affected. If one parent has trait and the other has anemia, that is sickle cell anemia, then the chances 50% will have, 50-50 will have. So 50% will have trait and 50% will have anemia. Okay, so all the children are affected. Again, if both the parents have trait, also sickle cell trait, what will happen? Then the chances are, there will be 1 in 4 chances that the child will be unaffected. So 1 in 4 chances is, this will be a normal child. How it becomes normal child? Because AS, trait has AS. So the AA will form, what will form? Adult hemoglobin. Adult hemoglobin is normal. So these are normal. But the other 2 will have traits and the last will have anemia. If you remember the genomic printing done by Mendel, you will understand this inheritance pattern. Okay, so homozygous and heterozygous has been done. Now what is double heterozygous state? Double heterozygous state means, you need to see that here the parent. Okay, if we see here, the common denominator is a sickle cell trait. All have sickle cell trait. Okay, so in here if we take the normal as sickle cell trait, that's for example, if we take normal as sickle cell trait and the abnormal as number 1 in beta thalassemia. If the father has beta thalassemia and the mother has trait, in this what will happen? The first kid will have beta thalassemia trait. Second child will have sickle cell trait. Third child will have sickle beta thalassemia disease, and the last will have no trait. Okay, so if you can make the inheritance pattern, you'll come to know how it is. Okay, I'll make this and I'll send you a photo of it. Okay, again the same thing. If you have hemoglobin C trait, in this what will happen? The first child will have hemoglobin C trait, then sickle cell trait, then sickle hemoglobin C disease and no trait. Okay, hemoglobin D trait, if you see, hemoglobin D trait will be there, sickle cell trait will be there, sickle cell hemoglobin D disease and no trait. Okay, so this is double heterozygous state. Okay, now coming to, first we are discussing heterozygous state. What is heterozygous state? It is also known as sickle cell trait. Here only one abnormal gene is inherited. Okay, one abnormal gene is inherited from a father or a mother. Okay, the patient with sickle cell trait develops no significant clinical problems except when they become severely hypoxic and may develop sickle cell crisis. Okay, so these are causes for hypoxic crisis. What happens when you travel to high altitudes, in poor health and in poor altitude? Okay, carbon monoxide poisoning, strangulation or smothering, very low blood pressure, smoke inhalation, choking, heart attack, medical condition, allergic reaction, severe cases of asthma, allergies, an infant due to improper sleep position or unsafe sleep environment, hyperventilation. So these are your hypoxic crisis. In these conditions, heterozygous state patients who have sickle cell trait will develop clinical symptoms. Okay. Laboratory findings for heterozygous. Here what happens? Patients usually do not have anemia because their red cell appears normal. But in hypoxic crisis, red cell crisis can develop. In hypoxic crisis, when you have red cell crisis, we do certain type of test. First is demonstration of sickling. The sickling is done. How it is done? This is the procedure. I have given a video which you can see. I'll just try to explain. Here what happens in a test tube, in this test tube we have to make a solution. How we make a solution? We take NS and we take sodium metabisulfite. Okay, we make a solution. After that we add blood. Okay, after we add blood we wait for 15 to 20 minutes. After 15 to 20 minutes we have to put on a paper or in a slide. When you put on a slide, you have to cover it with a cover slip and you have to let it rest. After you let it rest, you can see this kind of center formations. Okay, when you have sickle cell anemia and sickle cell trait, you can see this. You can see center very dark red color. Okay, but in a normal hemoglobin, if you check adult hemoglobin, this is very pale. Okay, now second is hemoglobin electrophoresis. In this what happens, you take a gel agar and you place inside the electrical chamber. Okay, and you add buffer solution to it. And these are some wells. In these wells, you'll put the samples. So this is a normal adult sample you put. Then you put a patient who you suspect to have sickle cell trait. Then you suspect another patient who can have sickle cell anemia. So you put in this well and you pass a current through it. In this current, what happens? From positive to negative it will flow. Okay, from anode to cathode it will flow. And in this what happens? If you see the normal hemoglobin will come and stop over here. Okay, in sickle cell trait, you'll have two band formation, and in sickle cell anemia, you'll have one band formation which will surpass the normal adult hemoglobin. Okay, HbA will be surpassed by HbS. So I have put a demonstration video of sickling which you can see. This diagram I've made a little big so you can see. So what happens is, you add blood to the test tube. Here the buffer solution is your sodium you have sodium metabisulfide and Na solution. This Na solution with the blood is, we have to wait for 10 minutes. After that we put that on a glass slide and then we cover it with a cover slip and again allow it to stand for 15 to 20 minutes, and then we can see the sickling that occurs. Okay. Then hemoglobin electrophoresis, I've also added a video, you can have a look. So you can see what happens is, after seeing the video also you can understand this. So what happens is, you pass the electricity from the anode to cathode. During this, what happens, whatever you filled in the wells will move because of the electrical currents. So what happens is that your HPA will form a single band, but it will stop over here. Okay. And your sickle cell anemia will also form a single band, but it will surpass, you can see it surpass the level. In sickle cell trait you have two band formation. So one band is suggestive of sickle cell anemia, two band is suggestive of sickle cell trait. Okay. Now coming to homozygous state. Homozygous state, you see the sickle cell anemia. Sickle cell anemia is homozygous state of HBS in the red cell in which an abnormal gene is inherited from each parent. So from each parent, so father, mother, you have one abnormal gene. Okay, so one abnormal gene, AS, one abnormal gene is coming. One abnormal gene is coming from both parents. Okay, so here what you see, if you see a normal RBC, it's a biconcave shaped oxyhemoglobin. This oxyhemoglobin, when it gives off oxygen in a deoxygenated state, it will still flow in the biconcave shape. In this biconcave shape, it will flow into the bloodstream. Okay, but what happens in sickle cell anemia? In sickle cell anemia, when an sickle cell RBC gives off oxygen, goes into a deoxygenated state, here your RBCs will become sickle shaped. How it happens is, if you remember the monomer, okay, you remember how the polypeptide chain was, you have alpha 2 beta 2 chains. In this, what happens, whenever oxygen is given in a deoxygenated form, it will start clumping together. This clumping together will form a sickle shaped RBC, which when flows into the bloodstream will cause obstruction. Okay, it will clump together and will block the arteries and vessels. Okay. Now what is the pathogenesis behind sickle cell anemia? First point is your basis of molecular lesion. In HBS, the basic genetic defect is single point mutation. Okay, in amino acid out of 164 in a hemoglobin molecule. So in a hemoglobin molecule, you have 146 amino acids. In that, one amino acid is changed. Okay, there is point mutation of one amino acid. The substitution of valine for glutamic acid at the 6th position of beta globin chain. Okay, which produces hemoglobin alpha 2 beta 2 S. Okay, so I do not know how to put the alpha and beta simple, so I've just put A and B. Okay, so alpha 2 beta 2 S. Okay, this alters the solubility of hemoglobin. Mechanism of sickling. Okay, mechanism of sickling. Due to point mutation, which leads to altered solubility of HBS, this will create a sticky patch on the surface of the hemoglobin S. So this hemoglobin that we had seen in the previous slide, this hemoglobin that we have seen, this hemoglobin will have a sticky side. Okay, this sticky side is present in both deoxygenated and oxygenated state. Okay, so mechanism of sickling, there is sticky patches on the surface of HBS, which is seen both in oxygenated and deoxygenated state. Only during deoxygenated HBS, the complementary sites for the sticky patches are expressed. The complementary site of deoxygenated HBS will bind to sticky patches of other HBSs. Okay, and this will start polymerization of hemoglobin to form needle-like insoluble crystals or rods of fibers within the RBC cytoplasm. These elongated fibers align and distort the red cells into classic sickle shape. This will distort the shape of the RBCs into crescentic or holly leaf or sickle shape or boat shape, These are known as sickle cells.